inviting me, particularly the team at Mātai, and I thought I would share a little bit of research from our group on pediatric biomechanics. So a special mention to my colleagues Julie Schwann, and you've just heard from Jeff Hansfield, and a lot of what I'll share with you today is from their work and their students' work. So what I like about this image taken from a marae several years ago is it first of all shows a group of kids potentially having fun here without any devices, which I quite like. Of course, our kids are dominated now with devices, and it has implications for their bone and joint health, as I'll discuss. The other thing is we see a cohort here of about 15. Now, if you look at the literature on pediatric biomechanics, the word population doesn't really come up very much, but the term maybe cohort does, and typically a lot of the work that's being done is on very small numbers, and so we think of a population of children, in this case, at least with pediatric biomechanics, as being about 10 or 15, which is actually not a lot, and we're hopefully going to change that. So we're going to start this talk about pediatrics. Of course, pediatrics is children, tamariki, and we'll start the talk with some pictures of bones of old people. So here are 3 females, and this is a mid-shaft CT scan, a micro-CT scan through the femur. And what you can see here, now these 3 individuals are very close in age, 78 to 80 years old, but you can quite easily see very different morphology of their bone. So you can see on the left a very healthy looking femur, nice thick cortex. This is the thick outer shell of the bone. And on the right, a bone that's osteoporotic. Now, clearly there's a difference between these two. And the question is, well, what drives this difference? And I'll let you read this quote, but it's from colleagues of mine, Dennis Carter and Gary Beaupre, who from Stanford University for years developed an area of research called mechanobiology, understanding how the mechanics of the tissue change the biology and vice versa, the two interchange. And what's really important here is to acknowledge, of course, that there's a biological component, the genes, and there's an outside environmental component that gives rise to this different in size and shape. And of course, that's mechanical forces. And so this is not new. I mean, in the 1800s, Julius Wolff, who was a German physician, nicely characterized the trabecular structure, the lines that you can see through the bone, and he indicated that those lines align with the principal stresses and the mechanical forces that go through the bone. So this knowledge has been around for some time, that mechanical forces regulate our different tissues. And of course, with bone, we can see that, but it's also inherent in the other tissues of the musculoskeletal system as well. And we've just heard a really nice talk from Jeff about fibrocartilage and fibrous connective tissue. Depending on the stress-strain history, the amount of pushing and pulling of these different tissues, you can change those tissues from one lineage to another. So you can turn bone to cartilage, cartilage to bone, and vice versa. So the mechanical forces really regulate the tissues in the body. Now this is important for us if we're thinking about engineering, because we want to understand the mechanical environment of this tissue. If we can do that, then we have a better opportunity to understand injury, recovery from injury, growth and development, as well as disease processes. And that's really what our kind of research group is all about, trying to understand this really intimate form-function relationship that exists in the musculoskeletal system. Now, of course, prior to Julius Wolff doing his work in the 1800s, this whakatauki really demonstrated that knowledge quite nicely. And of course, this is a public health message. So tama tū, tama ora, tama noho, tama mate, and we've already heard the term mate being used previously, and of course this is a common sense proverb. You know, an active child remains healthy, yet a lazy one becomes sick. And this is so true of the tissues of the musculoskeletal system. So again, this is an example here with bone, and again we're seeing the cortical bone, the outer kind of thick layer if you go through a long bone. And on the left-hand side, you can see nice, thick, healthy bone, and this is from a 27-year-old. As we age, we tend to see this bone change in its morphology, and what happens is the thickness changes, and it changes from the inside out. The inner cortical bone actually erodes away, which is kind of shown in the green line here, and you also see holes start to appear in the cortex, in that structure as well. And so this is an aging process. What we do know is that bones respond to the mechanical environment. And actually, if you look at this relationship here, there's a kind of a zone here. We kind of think of this as a lazy zone. Some sort of mechanical stimulus and the bone is happy. If you provide more stimulus, you get a response. And it's usually a positive adaptation for bone. You lay down more bone or you change the size of the cortex or the structure. But what's important to note is this little kind of cliff here. And tissues, bone as well as other musculoskeletal tissues, respond much more to disuse than overuse, which is over on this end of the spectrum. So unfortunately for us as a human species, we're more likely to be in this, all of us sitting around today listening to me, in disuse than we are overuse. And this is a major problem. And why is it a problem? Well, there is an epidemic really of bone and joint disorders, and that epidemic is on the rise mostly because of our physical inactivity. The other key thing to note here is that when we lay down the structure of our bones as well as other musculoskeletal system, but bones in particular, the juvenile skeleton during this period of adolescence is where we lay down the most bone, and it's where the mechanical forces have the most opportunity for strengthening the bone. The outer size of the cortex stays with us for the rest of our life. So what we do during growth and development sets up the skeleton for what it looks like for later in life, which means we could prevent bone and joint disease by doing the right thing during growth and adolescence. And I'm sorry, for most of us in the room, it's probably a little bit too late for that. Of course, you can delay the degenerative kind of changes as well by maintaining physical activity. So let's just think about pediatric bone and joint disorders for a moment. I'll talk about the three kind of main ones. Cerebral palsy is a neurological disorder, and the neurological disorder you could think of as stroke at birth. It affects about 2 in every 1,000 births. So 2 in every 1,000 infants have cerebral palsy. It's a neurological disorder, but what happens is the abnormal muscle forces that derive from the neurological condition lead to a worsening changes in deformity of the musculoskeletal system, and this worsens with age. So we do quite a lot of work on understanding the adaptations and changes of the structure with cerebral palsy. Another one that we're doing a little bit of work on is developmental dysplasia. The occurrence of this is much more common, although there's only a smaller percentage of those infants that have a developmental dysplasia that require some sort of intervention. And it's where the hip joint is not properly formed. This can give rise to chronic dislocation, which is obviously an issue, being physically active. And this last one here is a bit of a mouthful, slip capital femoral epiphysis. We'll have a bit of a test on this later on. SUFI is the common name for this. And the surgeons think of this as an ice cream falling off a cone, but the ice cream is the head of the femur in this case, and you can see the growth plate here. And abnormal or excessive loading applied to the head of the femur can give rise to this movement of the bone across the growth plate. Unfortunately, in New Zealand, we are as much as 8 times more likely to see this in our children than in other populations. And one of the reasons for that is our prevalence of childhood obesity. And so excessive loads during growth and development can cause such a disaster like this. And unfortunately, this child will need a joint reconstruction at a very early age. Okay, so what can we do about it? Well, if we try and understand from research perspective and look at the literature, you'll see that most research in this field is done on the aging skeleton, the old people in the room. No offense to the old people out there, and I call myself one now. Osteoarthritis and osteoporosis really drive the research domain. There's much, much less known about the growing skeleton, which is why we're doing our research. And of course, the longer term, if you think about it, the long-term consequences are much greater for a child who has a disorder compared to an adult. So it's really important that we do this work. So one of the projects we started several years ago is called the Musculoskeletal Atlas Project, or MAP. And the idea is to understand these structure-function relationships that occur in the musculoskeletal system. And we wanted to create an open source database that has a lot of information about this structure function. So one of the main sources of imaging data we've had is from the Victorian Institute of Forensic Medicine in Melbourne, Victoria, where we have access to several thousand full-body CT scans, And we have actually a particular cohort here with 333 of those several thousand that we have are from children. So we have age 4 to 18. And what we can do with those is we can create statistical representations of the morphology. So I mean size and shape. And we can do this to understand size and shape variations of bones. We can do this also to understand size and shape variations of muscle. Now, Jeff's given you a nice presentation about the connective tissue, and he's also done some work here with another one of his PhD students and working with Mātai to use MR imaging, which is particularly good for looking at soft tissue, to generate then populations, ideally longer term, and this is where the Tairawhiti study will be very useful, longitudinal population studies to look at growth and development of things like skeletal muscle. And this is an image here of a calf muscle, the soleus muscle in this case. Now we've done some of this work already looking at things like, well, okay, if the bone changes size and shape, you can also see the cortex or the thickness of the bone changes as well. And here the bone is responding to its mechanical environment and maintaining its stiffness, maintaining its strength. And the colors that you see changing here correspond to the thickness of the bone. So as the bone changes its shape, you can see it changing in thickness. And there's three kind of what we call modes of variation here that correspond to about 95% of the population. And you can see that here in the femur and how those modes kind of change. You can see some twisting. Some of these are sex variants as well, so they're more likely to describe a female femur compared to a male femur. And what we're doing now then is creating these population atlases of pediatric bone, which have not actually been shown before. So we can show the changes in density as well as shape and size of a range of different bones. And again, this is some of Jeff's work and his student Salim looking at muscle architecture. The colors you can see here from a child with cerebral palsy, and the colors indicate a deviation away from normal. And so the purple to black shows you 2 to 5 standard deviations away from a normal child who would have a healthy volume of muscle. And so by using these types of information, we can then classify and characterize what's normal growth and development, what looks healthy, and what is pathological. And that's useful for a surgeon who might want to classify some sort of pathology, but really we need to go a step further and understand its function. And that's really where then the work at the Bioengineering Institute has kind of pioneered, and you've already heard from Peter Hunter this morning on this, where we bring computational models in to understand what we can see and measure in terms of physiology. So here is kind of a standard, I guess, workflow where we take some medical imaging, some nice MR imaging here of the knee, we characterize some material properties of the tissues, then we do some experiments to understand motion, for example here kinematics, as well as potentially then the forces that cause that motion. We can use this then to characterize the localized stresses and strains of the different tissues, and this is really useful. But we want to bring this into the clinic, and there are challenges to clinical translation here. One of them is that it takes an incredibly long time to make these models, so we want to kind of automate that process. Another challenge is the models need to be tested and compared to experimental data. So we need to do a very good job of validating these models and showing that they're also reproducible so other people can use them. And the third challenge is that the models need to be able to predict some sort of clinical outcome. And so a lot of the work then that we're doing now is taking these population-based models and automatically creating workflows that can go into a clinic. So now we can automatically generate these very complicated finite element models just from some imaging data and our population-based models. So that's terrific. That's actually now part of some software that we're actually rolling out and expanding. And the last kind of example that I have here is on how we can use this in a clinical setting for a case of hip disorders. So one of the things that a surgeon might do if a child has a hip disorder is cut the bone, do what's called an osteotomy, and relocate the joint, reorient the bone. Now, wouldn't it be nice if you could virtually do that? And that's exactly what we've done here. We can do a virtual osteotomy. We can do predictions of how that changes the stresses throughout the cartilage and hopefully prevents the need for long-term joint reconstruction and saves the native tissue. So this is just an example then of what we're doing in the real world, also developing three-dimensional cutting guides for surgeons to be able to deliver the surgery in a low-cost manner, which we've also heard about today. So I'll close up some take-home messages. Loading your bones and joints during adolescence will prevent disease later in life. This is a public health message. Our image-based biomechanical models can be used to understand function and improve treatment strategies, and ultimately use it or lose it. So you've got to be able to kind of run out of here rather than walk. And I believe we have a bit of a break now, so do some star jumps on your way to grabbing a cup of tea. Acknowledge all the wonderful people involved in this work. Again, thanks to Mātai for inviting me, and here's a call out to all my acronyms in my talk. Thanks for listening.